All right, just want to start today, a little plug sent out by the pre-class academic advising center for the test course that they offer every spring. So those of you planning on taking these professional, the MCAT or the dentist's test, whatever, the DAT, if you're planning on taking one of those, you should prepare for it. So ideally, you're going to take a little lighter load next semester if you're planning on taking that in the spring, and take some kind of test prep course. Here is one option. This is only like $60, and you go once a week, and you brush up on all your science classes. Another option is taking a Kaplan course. You're running into some money to do that. It is very good, though. I can attest to that just from experience within my family. You can apply for a scholarship for that Kaplan course. The scholarship application is due in December 1st. So kind of think that way. If you're planning on going to professional school and you're not plugged in at the class advising center, then you want to do that soon. All right, and if you want me to send you this email, I can do that as well. So just a little plug for those of you that are thinking that way. Okay, the exam from Thursday went much better for a lot of you. Not for everybody, you always have some variation there, but the mean overall was a 78. So that's why I didn't curve it. Because we did have overall a little bit nicer distribution there. So some of you have come in already to take a look at your exams. I would encourage you to do that. I think that's helpful. A few more people to take it, hopefully today, and then hopefully we'll go over a few questions next class. So that's exam 3. All right, so we're going to pick off where we left off last time. We had started chapter 15. That's the chapter on innate immunity. We started out with that first line of defense, which is mostly talking about, barriers that we have, like our skin, defenses on our mucosal membranes. We talked about the role of our normal microbiota and how we think that's important to keep our immune system primed. And just some other, just bodily functions that serve immune functions that you might not have thought about before. And then when we organized it into our second line of defense, meaning when those first lines are breached, then we started talking about some of the cells that are important in our bloodstream, like our granulocytes, our agranulocytes, and some of their roles, which leads us into talking a little bit more specifically about phagocytosis. Okay, so we've talked about this kind of briefly in the past, but I want to kind of pick apart this process a little bit more carefully. All right, so the first thing that has to happen when our phagocytic cells are going to actually engulf some kind of invader is that they are going to follow chemical signals to the site where the invader is. All right, so we call that chemotaxis. They're going to follow cytokine signals. We'll talk a little bit more specifically about some examples of different cytokines in a little while. But again, that's movement towards the site of either injury or some kind of invader. Okay, and then we kind of skip over this step sometimes, but you have to remember that that phagocytic cell has to actually recognize that that invader is worthy of attack. Okay, it has to have a way to recognize this is not a self cell. This is some kind of invader. Okay, so the mechanism that our phagocytic cells have to do that are called toll-like receptors. Oops. Okay, and that's kind of a odd, it's not something that sticks in your memory, that term. Toll is a receptor that was originally recognized in fruit flies. And so when we realize that humans have something similar to that, that's why we call it toll-like receptor. And toll is a German word. So again, it's not an intuitive name, but we often abbreviate them and call them TLRs. These are proteins on the outside of our phagocytic cells. And remember, our phagocytic cells, that's primarily the neutrophils, the macrophages, and the dendritic cells. Okay, those are the primary cells that we have that do phagocytosis. They're all white blood cells. Okay, and I have your book does not show a graphic of toll-like receptors, so I'll show you one in just a minute. So it's always better to have a picture to work with. What do these toll-like receptors do? They actually recognize broad-based patterns that many pathogens are going to have. All right, so they're a little bit specific, but not terribly. When we really talk about specificity, then we have to get into our antibodies and our B cell receptors and our T cell receptors. These toll-like receptors are just recognizing motifs that many pathogens are going to have. So we call them pathogen-associated molecular patterns, or in other words, PAMPs. It's kind of a strange acronym. So some examples here would be things like LPS, lipopolysaccharide. That's something that's on the outer membrane of gram-negatives that it's never found in our bodies. Peptidoglycan, again, something that we as humans would never have. Maybe something like flagellin, that's a protein that makes up flagella. All right, just broad-based patterns, again, of things that would never be found normally in our bodies. And when the toll-like receptors recognize these patterns, then what happens? Well, that signals to the phagocytic cell, yes, this is something worthy to be phagocytized. But also, it's kind of like tripping an alarm. When those patterns are recognized, not only does phagocytosis occur, but then other aspects of the immune reaction are going to be called in. So increased inflammation, maybe some complement protein activation, which we'll talk about next. So other aspects of our innate immune response, inflammation, sending out cytokine signals, so other aspects can help out. All right, so again, this graphic is not in your book, but I think it's always helpful to have a picture. We've discovered somewhere, I think there might be 12 of them now in humans, but at least 10, we'll say 10 different toll-like receptors in humans. And you don't need to memorize, oh, TLR4 recognizes lipopolysaccharide. That's not necessary. Incidentally, the people who discovered that, I think in the early 2000s, got a Nobel Prize for it, though. So this is, fairly recent discoveries of toll-like receptors. But there's some in again, in the outer, in the cell membrane, the outer portion of our phagocytic cells. Here's, say this is a macrophage, for example. Recognizing these broad-based patterns like LPS, flagellin, various membrane of cell wall components, not just a bacteria, but maybe yeast or, not just gram-positive and gram-negative bacteria. And so we also will express some toll-like receptors on the endosome. So when the ones on the outside are going to trigger, yes, this pathogen is worthy to be ingested. And then within that membrane that it's phagocytized in will express other toll-like receptors, and those are going to recognize parts of a chewed up cell that are recognized not as self. So things like double-stranded RNA, that's not something that we would ever find in our cells. Other aspects of nucleic acid that might be a little bit different than our nucleic acid, like what's methylated, or just kind of more subtle changes, differences in viral and bacterial nucleic acids. So obviously those toll-like receptors are going to, when they're recognizing those PAMPs, that's going to, again, alert other aspects of the immune system and increase the innate immune response. All right, so if we look at the steps of phagocytosis, which we've done so very briefly in the past, this is from your book now. It starts out with chemotaxis towards the microbe. And they attempt to kind of show you in a transmission electron micrograph what that might look like. And then they kind of draw the rest of it so you can really tell what's what. So the macrophage moves towards the microbe. Secondly, it adheres to the microbe as well as recognizes it. Your book kind of glosses over that in this picture. And then I want to just point out that what helps the phagocytic cell adhere to the microbe, anything that helps with that process, we call an opsonin. Okay, so that's probably a new term, one that you'll want to know. An opsonin is any factor that enhances phagocytosis, or in other words, makes it more likely that the white blood cell is going to be successful in phagocytizing that microbe. And we just got done talking about in chapter 14 how microbes have virulence factors that would make it less likely that they would get phagocytized. They might have a capsule, for instance, that makes them harder to be phagocytized. Our white blood cells have tools, or we could say our immune system has tools to make our cells more likely to be successful. Things like complement proteins. And we're going to talk about that next, and how that will improve the phagocytic cell's chances of grabbing hold of that microbe, as well as antibodies. All right, so antibodies can act as an opsonin. If they are bound to that, say, bacterial cell, then those pseudopods are going to be more likely to be able to grab a hold, like the handles on the microbe. Okay, so we'll revisit both of those because we haven't talked about either one of those things yet, but they are considered opsonins. And then ingestion. All right, so pseudopods are extended. That microbe or invader is going to be surrounded in a membrane-bound vesicle, which we call a phagosome. And then the phagosome eventually is going to fuse with the lysosome. So lysosomes, that's an organelle that contains lots of digestive enzymes. We're going to look at some examples of enzymes that's in there in a second. Where did the lysosome come from? All those enzymes that's in there get manufactured in the rough ER, so the ribosomes on the endoplasmic reticulum, and then they're packaged up in the Golgi, and then they are in the cell and will, fuse with that phagosome. Let's look at some of what's inside of a lysosome, and then we'll come back to this graphic in just a minute. There's actually about 40 different proteins that we have figured out that can be in our lysosomes. So just some examples of the kinds of things that are in there that are going to usually be able to kill any pathogen that's in there. Lots of digestive enzymes. So enzymes that degrade proteins, proteases, enzymes that degrade lipids, lipases. Lysozyme, we've talked about that. That's the one that breaks the bonds between the sugar subunits of peptidoglycan. Enzymes that break down nucleic acids. Lots of other antimicrobial proteins. And these are actually all going to work best at a low pH. And within the lysosome, there are mechanisms to pump in protons to lower the pH. Okay, so that's another mechanism. Yeah, the low pH is likely to be detrimental to the pathogen, but all these enzymes are working best at that pH. When the lysosomal contents are eventually exocytosed or transported out of the cell, okay, all those enzymes aren't really going to be detrimental outside of the cell because they're not working as well at that more neutral pH outside of the cell. A couple other examples of enzymes that I wanted to mention, they're considered respiratory burst enzymes. Okay, so these are really cool. Actually, within our lysosomes, we have enzymes that on purpose produce toxic forms of oxygen. Okay, so things like superoxide ion, hydrogen peroxide, hydroxyl radicals. Okay, and remember, those are stealing electrons from lots of different molecules, so they contribute to the damage caused to the pathogen. And interestingly enough, in our neutrophils, we have an enzyme called myeloperoxidase. And that's an enzyme that can actually make hypochlorite from hydrogen peroxide and chloride ion. Okay, remember what hypochlorite is? That's bleach. All right, so within our neutrophils, we're actually bleaching pathogens. So that's kind of cool to think about and amazing that we can do that, really. Okay, so the upshot is most of the time, 99% of the time, the pathogen is going to be killed within that phagolysosome. Okay, every once in a while there are different adaptations that bacteria have, like the Mycobacterium tuberculosis. I think we mentioned this briefly. Will keep the lysosome from fusing with the phagosome, and then it lives, quite happily within that phagocytic cell. But as long as the lysosome fuses, usually the pathogen's going to be killed, and then exocytosed outside of the cell. One other thing to mention about this process, and I can't resist the analogy here because I'm a child of the 80s, is that there are different levels of activation. Okay, especially, we know this is especially true with macrophages, because macrophages will exist kind of at one level where they're patrolling around the body, and remember, they live a long time and they're just going around eating up garbage, basically. So they're chewing up old cells that are dead or dying and kind of getting rid of them. Well, they can actually function on a higher level of activation when they get the appropriate signals to do that. When they're in the neighborhood of some invader, again, if they get the right cytokine signals, they can become activated. So what does Pac-Man normally do? He goes around and eats up the dots, right? So he's eating up, say, the dead cells. But when Pac-Man swallows one of those big dots in the corner, now Pac-Man is bigger and he moves faster, and now he can not only eat dots, but he can eat the ghosts as well. All right, so that's exactly like macrophages. Once they're activated, they get a little bit bigger, they can phagocytize faster, they are going to produce more lysosomal proteins, overall increase their metabolism, and make even more cytokines. So they're even more likely to be able to kill invaders. And it does come at some cost, though, because when you produce more cytokines like that, it's likely to cause a little bit more inflammation and maybe actually result in a little bit of tissue damage. But that can be temporary, and at least hopefully the pathogen is conquered. All right, and let's see if I can get to this video, but this is the best video that I have ever seen of macrophages in action. Let me see if I can bring this up. Okay, I probably won't have sound here, but this is just a short clip.
Here, what you're looking at is a video of macrophages that have been given yeast to play with, and what you can see is the macrophages will move towards the yeast.
Of macrophages this is not real time, it's you know, they've sped it up. They're not actually moving this time lapse, is that what we call it? They're not actually moving that fast. But he said that they've added, these are macrophages that have been activated, and they've added some yeast to the medium. So the macrophages are going around, they move by amoeboid movement, and they are swallowing up some of the yeast.
And then engulf them.
Okay,
so here one is moving directly towards it and then engulfs it.
Like right
Here's
here.
another yeast that's here, which will eventually be attacked and eaten by another macrophage, shown right there. So in much the same way that Metchnikoff found, these invading microorganisms were activated the macrophage, turned it on, increased its capacity for migration, its capacity for cytotoxic killing, and as shown here, its capacity for actually killing and eating the invading microorganisms. This is
So I posted that link. It's just the beginning and it goes on, so show some other things that aren't as interesting to us. But all right, so that's phagocytosis. Okay, another aspect of our second line of defense are complement proteins, and I just mentioned those briefly as they can be important opsonins. Okay, so I want to just kind of talk about a little overview here first, until we'll get more specific about what they actually do, and I'll show you your graphic from your book here. So complement proteins, what are they? They're a series of about 20 different proteins that normally we have in our bloodstream. They're actually made in the liver. And what they do is complement, or maybe we could say help, other aspects of innate immunity. And they also have the capacity to actually kill invaders as well, certain kind of invader. Okay, so they complement other aspects of immunity. Part of our innate system, and again, we consider them part of innate immunity because they're going to have the same reaction like every time that invader is encountered. Okay, it's not going to be a better reaction the second time. It's just the same every time for the most part. These proteins, we call them, it's a cascade reaction because actually one protein will act as an enzyme and split another protein to activate it. And then those two bits will split other proteins. So it's kind of like one protein acts on another, acts on another, acts on another. So that's why we call it a cascade reaction. What do they actually do? Some of these proteins act as opsonins, and I'm going to illustrate that shortly. Some of the complement fragments can act to increase chemotaxis of cells and inflammation. And I know we haven't talked about inflammation yet and what that is exactly, but we will shortly. And some of these complement proteins can act to form what's called a membrane attack complex. Okay, we refer to those as MACs, membrane attack complexes. Basically, some of these complement proteins can act to form a pore in a bacteria's outer membrane as well as cell membrane. Okay, so they can act directly on invaders to kill them as well. Okay, so here's a little overview of how these proteins work. They can be activated in several different ways, and we'll get to that in a minute. But one way that they're activated is classically, and that just involves an antibody and an antigen bound together. And we'll talk more about what both of those things are, but basically we have antigens on the surface of, say, a bacterial cell. So something on the surface of a bacterial cell that would elicit an immune reaction, and an antibody bound to that antigen will be recognized by the first complement protein, which is right here. And then that complement protein acts on other complement proteins, so on and so forth. Some of these bits will act directly to increase inflammation, increase chemotaxis of cells, and increase the chances of phagocytosis, act as opsonins. And some of the fragments will form this cylindrical complex in a bacterial cell wall and cell membrane, poking holes in it, which can bring about the death of the bacterial cell. Okay, so that's your little overview. Here's the graphic that your book is in your book. Okay, so this is one of the most common questions that I get asked. What do I need to know about this? So I've tried in your objectives to be really clear. Okay, make sure you know this, 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 and this about complement proteins. So you don't need to be able to draw this out, but let's walk through it so we kind of understand where these fragments are coming from and which ones do what. Okay, so this is again showing the classical activation where you have an antibody here that's bound to a bacterial cell. So when the antibody recognizes the antigen, if that C1, that first complement protein, is kind of in the neighborhood and it recognizes that, then the C1 will act to split complement fragments C2 and C4. So when a complement fragment splits, we call the resulting bits, in this case, C2A and C2B, and C4A and C4B. Okay, so when one of these fragments is acted on, it splits, and then the C1 actually acts as an enzyme on C2 and C4. Okay, some of these bits here will form up together, one from C2 and one from C4. You don't have to remember which is which. This C4B, C2A, actually. And anytime anything is really important, we like to give it another name. In this case, we also call this C3 convertase. Okay, these two bits, when they come together and act as an enzyme, they actually act on complement fragment C3. So I call it C3 convertase. And that splits C3 into C3A and C3B. Okay, now we're getting into bits that actually act and do something. That C3A here triggers inflammation. And we'll see in a minute how that's beneficial. C3B acts as an opsonin. So actually the C3B binds up here to the pathogen. Okay, B binds. Okay, and now a pseudopod would be more likely to be able to grab a hold of that pathogen, like from a macrophage, for example. Okay, some of that C3B binds back with that C3 convertase here and acts as an enzyme on C5 to split C5. And now we're getting C5A and C5B. So the C3A and the C5A, they act to increase inflammation. And part of increasing inflammation is increasing chemotaxis of cells. So I won't write that in. But if you think A, allergen, then that might make you think inflammation. So the C3A, the C5A, they both increase inflammation. I'm going to show you how in a minute. The C3B and the C5B, they are going to act as, they're going to bind, B for bind, to the bacterial cell. Because the C5B, we can see that up here, it binds to the bacterial cell along with C6, C7, C8, and a bunch of C9s. Okay, and they are making the membrane attack complex. Okay, so when they all come together, they can poke a hole in the membrane, the outer membrane of that bacterial cell, and actually form a pore. Okay, so what does a membrane attack complex look like in a real cell in a transmission electron micrograph? You can see all these little holes poked in the cell. Okay, one hole wouldn't make that much difference, but you get a whole bunch of them like that, that cell is in trouble. This is going to be most effective against what kind of cell? Remember, gram-positives have that really thick peptidoglycan. Okay, so membrane attack complexes are not going to be able to poke through gram-positives. They're going to be most effective against gram-negatives because they have an outer membrane, they have only a thin layer of peptidoglycan. So those membrane attack complexes not only pierce the outer membrane and the peptidoglycan, but also the plasma membrane. All right, so that cell's got problems. All right, a lot of cellular processes are disrupted when you get pores like that. So the membrane attack complex, again, that's going to work primarily against gram-negative bacteria. Okay, so there are three different ways that that complement cascade can be activated. So we talked about one way, that classical pathway. This involves antibodies. So as we'll talk about probably next lecture, antibodies are typically not produced till later on in the course of an infection because B cells have to be activated. They have to get signals from antigen presenting cells. So that takes a while. So that's not going to be the first way in terms of, in a course of reaction, infection, that complement's going to be activated. Typically, it's either the alternative or lectin pathway that might happen first in the course of an infection. The alternate pathway, if we were to kind of just simplify that, we would say that it's activated by pathogens. But we're going to look at that a little bit more closely in a second. And the lectin pathway is activated by polysaccharides on the surface of bacteria. So we'll look at a picture there that may help a little bit to remember those three ways of activation. This is a little YouTube clip I just watched again before I came to class. It's just kind of a little sum up of complement and everything it does. They talk really, really fast. So you might have to watch it a couple times, but it's only a couple minutes long. Again, it's a nice little sum up that might help. So here's the three activation pathways, the way complement will start to work. And I should mention, and what this graphic tries to make clear is no matter how it's activated, whichever pathway, you get the same result. Like you get the C3A and you get the C3B and the C5A and the C5A and the membrane attack complex. You get all those things no matter how it's activated. So it's not like, oh, the antibodies have this kind of complement and, you know, whatever, the other ways have another kind. It results the same, it just can get started differently. All right, so the classical pathway involves the antibodies. The alternative pathway, one thing to remember here, and let me make a note of it over here, is that that fragment C3, when that fragment is split, that's really when you start to see the benefits of these complement proteins. C3 is kind of randomly split at a low level all the time. Okay, so even with nothing activating it, C3 will just kind of be randomly splitting every now and then. Again, not very often. And if it happens, if that fragment that results C3b happens to be in the direct vicinity of a microbe, it will bind to it. And if there's other signals around, then it will basically activate the rest of the complement cascade. Okay, so I'm simplifying that a little bit. Where do these come from? Don't worry about it. If C3b is not in the direct vicinity of a microbe, then it just degrades, doesn't do anything. But if the pathogen is nearby, then that pathogen basically can activate more splitting of C3. And then the third way is called the lectin pathway. So bacteria very commonly will have a sugar on their surface called mannose. Okay, that's a sugar that they have on their surface, a polysaccharide that we don't have on the surface of our cells. We make proteins in the liver called lectins that recognize that mannose. Okay, so when lectins are bound to mannose on the surface of the pathogen, that will act to split C3 as well. Okay, so that can cleave C3. So we think that the alternative and the lectin pathway are probably the most common ways that complement is activated, but 3 different pathways. And all these aspects of complement, they would act on our cells as well, except our cells have lots of proteins on our surface just to downregulate complement from binding to our cells. So it's a pretty intricate thing. This is kind of the basics of it, but we do know that people that end up getting certain infections over and over and over again, they realize, oh, it's because you have a deficiency in whatever, some complement protein. Especially people that end up getting bacterial meningitis, maybe a couple times. It's a pretty rare infection. And usually if you get it a few times, it's because your complement is not acting like the way it ought to be. All right, don't be afraid to stop and ask me questions or to jump in here. All right, so I've talked about inflammation several times here. So finally, we'll get to talking about what it is exactly. There's a really great tutorial on inflammation that's part of your homework. So it's Dr. Bauman's tutorial on inflammation. It's 4.5 minutes long. It just explains everything that I'm going to explain right now. So don't watch that at like 6 times speed. You know, do that in your homework, pay attention to that. And I think that will be helpful to hear it again, for sure. I know that we're accustomed to thinking about inflammation as being a really bad thing, but in small amounts, inflammation is necessary in order for tissue repair, for one thing. Anytime we have some kind of wound, there has to be some inflammatory response before our cells can start to regenerate our tissue, even if there's not an invader present. And inflammation also helps to mobilize and attract lots of aspects of our immunity, immune cells, complement, etc., to the site of injury. Okay, so again, does have a very important purpose. Again, it can go on too long though. So oftentimes we're trying to dampen down inflammation. Hallmarks of inflammation. So how do we know there's an inflammatory response taking place? You've probably all experienced all of them. Redness, localized heat, pain. Okay, those increased blood flow can act on our neurons in the area of inflammation, as well as swelling. All right, we'll refer to that as edema. It's kind of a fancy word for swelling. All right, so here's an example of some inflammation taking place. This is a, it's not an infection, it's a condition called chilblains, where you can get sensitized to cold, basically. So even though this person hasn't been out, you know, in the snow or whatever, maybe just, you know, when you wake up in the morning, you can have this inflammation in your toes. So you can see the redness. it looks like it would be hot to the touch. It looks painful. It looks like there's a little swelling of the toes as well. I had my 3rd booster shot last week. All right, so I experienced all these things, a little redness around the site of infection. It was warm to the touch. It hurt a little bit, not too bad though. It was a little bit swelled up. So lots of times where we have inflammation. This can be acute. So, you know, it lasts a while and then it resolves. The problem is this can be chronic. I went to the doctor last weekend because I've been having trouble with my hip flexor, which it turns out is one of the big tendons in your body. The doctor said, we have tendonitis. That's inflammation of your tendon. He's like, how did you do that? And I said, well, I was doing hurdle drills 7 months ago. And he laughed at me. So what are you doing hurdle drills for? I said, well, I'm a track coach. Don't laugh at me for doing that. But anyway, I didn't warm up when I was doing that. So he said, well, you've got inflammation there. You have to take anti-inflammatories to get rid of that because now it's chronic and that inflammation is not doing you good anymore. So it's very common for people to take anti-inflammatory drugs when things become chronic. All right, well, what triggers inflammation? So one of the things that can trigger inflammation, I'm gonna go back to those complement fragments. So C3A, remember said A, you can think of allergy, C5A. What those do in order to trigger inflammation, they actually bind to our granulocytes. Remember our cells that are granulocytes, all the phils, basophils, eosinophils, neutrophils. Those are our granulocytes, as well as mast cells, which we didn't really talk about, but those have granules as well. So those complement fragments bind to those granulocytes, and cause them to degranulate. I had trouble saying that word last class. Inside those granules, there's some digestive enzymes, also inflammatory mediators. All right, and I'm not going to go too deep into examples there. Probably the inflammatory mediator that you've heard of the most, such as histamine. Common to take an antihistamine if you have a cold. Okay, so something like histamine, what's that do? It's going to act on blood vessels. Okay, so next graphic, how do those inflammatory mediators act on blood vessels? Okay, well, they're going to dilate them. They're going to make them bigger. Okay, so after an injury or in response to a pathogen, what happens? Here's our normal state. So before you have some injury or exposed to a pathogen, maybe what the capillary bed should look like. When you have a release of these inflammatory mediators, so that's these little blue dots here in this graphic, you can see how it's making those blood vessels get larger. That means dilate. Okay, so vasodilation. Okay, blood vessels, they're going to increase in size. They're going to get larger. And they're going to get leaky. So basically the interstitial space between the cells is going to get a little bit larger. That's how they increase in size. Okay, so when you first have an injury, you actually get constriction of blood vessels so you don't bleed out. But very soon after that, they're going to dilate to get increased blood flow to the area, because that increased blood flow is going to bring some of the cells that are going to bring about healing. Okay, so here's a close-up of those blood cells. Okay, so normal permeability of the venule here. So you can see these interstitial spaces, the spaces between the cells, it's pretty tight. You have a little fluid leaving the cells, plasma, and we'll see, maybe we'll get to it next time. Where does that plasma normally go? It gets picked up by lymphatic vessels. Okay, that normally happens. You've got your blood cells whizzing through your blood vessels pretty fast here, both your red blood cells, and this is a monocyte. Remember, those monocytes, when they move into tissues, are going to differentiate into macrophages and dendritic cells. Okay, so in the presence of some inflammatory mediator, you can see how the space between the cells gets bigger. We have a big arrow here of fluids leaving the cell. Okay, that's going to contribute to swelling. We have lots of fluids moving out into the tissue. But with those fluids are going to come with the fluids are the cells that need to get to where the site of the injury is. So here's the monocyte leaving the blood vessel. And that's actually a very complicated process of how that comes about. If you read about it, there's like 10 different signals that have to be met for that monocyte to actually do that. First of all, the blood's moving through there really quickly, so it can't just take a right turn and squeeze out through that blood vessel wall. It has to receive the correct signals to first stick to the blood vessel wall. And we call that process margination. Okay, so that's the process by which white blood cells adhere to the sides of the blood vessel. Okay, so it has to kind of slow down and eventually stick to the side of the blood vessel. And then the process by which it actually squeezes out through the blood vessel wall, that's called diapedesis. Okay. That's the process by which white blood cells leave, or we could say migrate, through the blood vessel walls. Okay, so first there has to be margination, and then there can be diapedesis. Okay, so that monocyte now is going to differentiate into, say, a macrophage, and it's going to move either by amoeboid motion, or it can be carried by those fluids, follow the chemical signals till the site in the tissues of where the invaders are. Okay. All right. So the effects of that we feel, coming back to that redness and heat, those are both due to increased blood flow. The pain that we feel, those inflammatory mediators actually will stimulate nerve endings. So nerve endings stimulated. Okay, and edema, what good does that do? Well, it helps dilute the effect of any virulence factors released by the pathogen. So if you got a pathogen that's secreting a toxin, for example, or an extracellular enzyme, if you try to dilute that out a little bit, that's going to be helpful to us. It might bring clotting factors to a site of injury, and certainly get the right cells to the area, like macrophages and neutrophils. That edema can last a long time. It actually gives the appropriate cells, maybe even lymphocytes, a chance to get to the site. And eventually that fluid, like I said, will drain into the lymphatic system, lymphatic vessels. Okay, so again, short-term, good thing. Usually though, it lasts longer than what we want it to. We get tired of it hurting, we get tired of the swelling. We might apply some ice to try to get rid of it. We might take some anti-inflammatory medicine that will all kind of dampen the effects. One other potential effect of inflammation can be fever. So let's talk about what good fever is. We're about ready to wrap up our innate immunity here. Fever is caused by pyrogens. All right, so that prefix pyro, that means fever inducing. Okay, so a chemical that induces fever is called a pyrogen. And sometimes a pathogen itself can act as a pyrogen, but usually the pathogen causes our cells to release cytokines that will act as pyrogens. And these are chemicals that are acting on the hypothalamus. So that's deep within our brain. That's our body's thermostat, basically. All right. So when chemicals act on that, they act as pyrogens and cause a little increase in our body temperature. What are the benefits of fever? I think probably the most useful benefit of fever is that it's a useful measure of health. Okay, so particularly for anybody that doesn't communicate well, say a baby, why is this baby screaming all the time? Oh, they have a fever. Okay, there's probably some infection going on here. And fever can be as a result of some cancers, but most often it's some kind of infection. So we know, hey, I need to stay away from people. Hey, I need to get some treatment maybe. In terms of helping us fight the pathogen, it can inhibit the growth of the pathogen. Often though, really just raising our body temperature up a degree or two is not going to make that much difference. For most pathogens, they have a wide range of temperatures that they're going to survive at. So that effect is probably going to be minimal. The one exception to that is back in the early 1900s, 1917, a guy in Germany realized he was trying to find a cure for syphilis. And syphilis is one of those bacteria that if you get a high fever, then that actually that will kill the pathogen. So he realized that to treat syphilis, he started infecting people with malaria because malaria caused a really high fever. And that would take care of the syphilis infection. That was before antibiotics, right? And malaria, you might survive malaria, but syphilis can be really bad. It can cause you basically all these neurological effects and eventually kill you. So he won a Nobel Prize for that. And that was the way we treated syphilis until antibiotics came out in the 1940s. But again, usually that's very minimal help. Not going to inhibit growth of pathogens very much. We do know that a fever will stimulate our immune system in other ways. It can basically, you have higher temps, you have faster reactions. So you have, better activity of immune cells, more release of cytokines, increased mobility of immune cells. So this is probably the biggest benefit that we get from fever. We know that it can also act as a signal to impede nutrient availability. Okay, let's see, especially in terms of iron. Okay, so when cells sense that fever, they get the signal to kind of take up any available iron around their cells. And iron is often a limiting growth nutrient for bacteria. So the benefits of fever are probably slight. So the question is, well, when you have a fever, should you treat it to bring it back down? Or is this a big benefit? You should let the fever rage, or should you treat your fever? It's controversial, but most of the time doctors say, go ahead and treat your fever because these benefits are fairly slight. And they don't outweigh the discomfort caused by fever. So practical application there. It's okay to take some Tylenol if you got a fever. All right, so we're almost through our second line of defense. We talked about cells last time. We talked about complement, inflammation, fever. One more thing I just want to circle back to briefly here is thinking about some of these signals that I've already talked about or mentioned briefly, and that's cytokines. All right, and this slide is exactly what they always tell professors: you shouldn't make PowerPoint slides like this. You shouldn't have all these words on them. But I just want to mention this briefly, and I just mostly want you to listen a little bit. Hey, these are examples of what cytokines are. Okay, so there's only one that I'm going to mention anymore, and that's interferons. So what are cytokines? They are signaling molecules that lots of our immune cells are going to use to signal all different other kinds of cells. The term that we used to use was interleukins. So we had IL-1, IL-2, there's like 33 different interleukins. But we realized that those signals weren't just acting between white blood cells. Leukin means white blood cells. That they are acting on all different kinds of cells. So we decided that wasn't a very good name, but we used it so much that it was too familiar to throw out. So now we just say, well, interleukins are a type of cytokine. Okay, so they're a signaling molecule. Another type of cytokine that you may have heard of is tumor necrosis factor, TNF. We call this cytokine tumor necrosis factor because we discovered that in mice, when you concentrate a bunch of it and inject it into mice with cancer, it made their tumor cells die or necrose. When we tried that in humans, it caused such galloping inflammation that it caused patients to go into shock. Okay, and I forgot to mention that when we were talking about inflammation. If our blood vessels get too leaky, that's a problem because we're not getting enough red blood cells that are carrying oxygen, and your organs don't get enough oxygen, your blood pressure goes way down, and that can cause shock, and that's potentially deadly. All right, so you get too much inflammation, that's really a bad thing. So we can't use this to treat cancer. Oftentimes, and this is just an example, just examples of what these things do. Don't memorize those. But you may have heard people that have autoimmune infections, some drugs like Humira, that's one that they advertise on TV all the time. It's actually a tumor necrosis factor blocker. All right, so people take that for Crohn's disease or rheumatoid arthritis to try to dampen the tumor necrosis factor. Again, our body's just making too much of it, and that can be a bad thing. All right, so I do want to briefly mention how interferons work. Okay, there's two different kinds of interferons. Interferon gamma is part of adaptive immunity. We're not going to worry about that. But I do want to mention what we call type 1 interferons, alpha and beta, because they're one of the only helps that our innate immunity has against virally infected viruses. Okay, so one of the only ways that we can treat virally infected cells. Okay, so I just want to look briefly here at how interferons work. And again, this is your figure from your book. It's kind of interesting because the cell that secretes interferon, it doesn't actually help that cell. It's going to help surrounding cells. All right, so here's an infected cell. It's been infected by a virus. The virus is taking over the cell. But the cell, in response, will make interferon and secrete it. And then neighboring cells will have interferon receptors. So this cell is not infected yet, but now it's ready. It's getting prepared for that viral infection because that interferon causes it to transcribe and translate these proteins that are called antiviral proteins. And they are just kind of hanging out inactive. Over time, this infected cell is going to die. Okay, eventually, again, the cell takes over, makes more virions, and it lyses or buds out of the cell. Okay, so those new virions maybe now at a later time are infecting this cell that originally was uninfected, but was prepared because it got these interferon signals. So now that the cell senses that the virus is infecting it, it activates those antiviral proteins, and those are going to act to stop protein synthesis. Okay, they bind to the ribosome and stops any protein synthesis in the cell. And you would think, well, that would be harmful to the cell. Yes, but the virus can't use the ribosomes to make its own viral proteins. So, you know, a cell can withstand not making its own proteins for a while. And meanwhile, in those 2 days, at least no new viral particles are being made. So it's going to slow the rate that viruses are going to be transferred to new cells, and kind of let other aspects of our immunity maybe take care of the virus. Okay. So lastly, I'd like to call attention to some of the tables that your book has. This is a nice sum up of innate immunity. Sometimes we get so bogged down in all the little details that we forget the big picture. So this is a good thing to kind of, cover up the complement and think, what does complement do? Or cover up interferons. What does interferon do? You know, we didn't really get too specific here talking about some antimicrobial peptides, but most of this we talked about. So just draw your attention there. Okay, so that's chapter 15. The homework here is a little bit maybe longer for chapter 15 and chapter 16. So I'd encourage you to start. You can start on that now and kind of chip away at it. There's lots of animations that are going to be helpful. So I encourage you to do that. So we'll move on here to the third line. Sorry, that got chopped off a little bit there. Into chapter 16 now, we're going to be thinking about specific immune responses mediated by B cells and T cells. Okay, so chapter 16, some of you have been asking me, how do I study this immunology stuff? I would really encourage you, if you haven't cracked your book yet this semester, to use that book now. Chapter 16 is kind of long, but we're covering most of it. And it's going to be really important to hear what I talk about in class, maybe a little different way, but very similar way in the book. So again, repetition is going to help you. It's presented in a little different order in the book, and that actually may be helpful as well. There's not really a right way or a wrong way here. So I would encourage you to use your book. Even like in chapter 15, what's that complement stuff? Okay, there's like a page of that in your book. So go back to your book and read through. It's not overwhelming. You can kind of pick and choose. But again, chapter 16, I encourage you to read your book. So what are we going to cover here? We're going to start with a little anatomy. So to begin with here, probably a lot of you are more familiar with that than I am. So we're going to talk about the lymphatic system. I've tried not to use this word too much, antigens, until we actually define it, but we'll be looking at what an antigen is and what makes a good antigen. And then we'll move into antibodies. I can remember always can't keep those straight, which is which, antigen and antibody. Okay, if that is you, then you're gonna learn the difference between those. This is the most complicated part here. How do cells advertise that they are infected by a virus? There's a specific mechanism that we have to do that. So we'll talk about that in preparation for how T cells and B cells get activated. Okay, so we're going to build up to that, and hopefully it won't be too bad. All right, so little overview here. Adaptive immunity, that's your body's way to respond to specific pathogens or distinct invaders. We have to have those innate responses in order to call up your adaptive responses. So it's the innate cell's duty basically to get those specific antigens presented to B and C T cells. All right, 5 attributes of adaptive immunity. So we could describe adaptive immunity, number 1 is having very high specificity. Our B cell receptors and T cell receptors will respond. We have B cell and T cell receptors that will fit almost any type of invader that you can think of. You probably have B cells that would recognize stingray venom, to use the example they use in your book. Will you ever encounter stingray venom? Probably not. Just in case you do, we are ready for that. Okay, so you have B cell receptors, T cell receptors that will recognize that. And then once they recognize it, then they can be induced. So inducibility, they become activated, and then they can make many more of themselves. So you probably only have a few cells that with that specificity, and then they'll make many more. So clonality, they'll make many more of that same kind of cell when we need it, only when we need it. Okay. Our adaptive immune cells should be unresponsive to self. Okay, we have mechanisms in place so that happens. That's part of the maturing of the B and T cells, that when they have receptors that recognize our self, then they're allowed to die, basically. So we have mechanisms for that. When those mechanisms fail, that's when we have autoimmune disease. And very importantly, adaptive immunity is characterized by memory, meaning the next time that pathogen's encountered, that we'll have a faster and better response to it. With our innate cells, there's very little learning that takes place there. You just have the same response every time that pathogen's encountered. So because we have memory with our B and T cells, that forms the basis of vaccines. And that will follow, that will be our next discussion after we get through adaptive immunity. Okay, so adaptive immunity involves the action of lymphocytes. Okay, so B cells and T cells primarily, remember we called natural killer cells lymphocytes as well, but they don't have much of a role in adaptive immunity. So B cells, they are responsible for antibody immune response. Sometimes we refer to that as humoral immunity, humors meaning bodily fluids. So we find antibodies in our bloodstream for the most part. They are going to act on extracellular pathogens. Oops. Extracellular pathogens. Okay, and we'll say and toxins. Okay, so anything that's outside of the cell, that's where antibodies are going to be working. Contrast that with something that's inside of the cell. T cells act on intracellular pathogens. Okay, so that can be there are some bacteria that will live inside of a cell or have to be inside of a cell, but mostly I'm talking about viruses here. Okay, so we have ways to alert T cells that our cells have been infected with a virus. And then what the T cell does is destroy that host cell. Okay, so our B cells, our antibodies can sometimes the analogy is, oh, the antibodies are bullets, but the T cells, they're the hand grenades. Okay, they're really doing a lot of damage. Okay, we call this a cell-mediated response. All right, so that will be more clear, the kinds of pathogens that they're going to act on as we move forward here. All right, so looking at where some of these actions and immunological synapses are going to take place, a lot of it is going to take place in the lymphatic system. Okay, so we have primary lymphoid organs. That's where lymphocytes are made. So that's the red bone marrow. Okay, that's where B and T cells are made. So red bone marrow, we find that in our kind of bigger bones, like our femur, our hips. Okay, that's where we're going to find that kind of red bone marrow. And B cells are going to also mature in the red bone marrow. So when I say mature, that means they're going to, their B cell receptors, we're going to learn to get rid of the B cells with B cell receptors that recognize self. Okay, so that's part of the maturation process. T cells are going to be made in the red bone marrow, but then are going to, they're going to mature. T cells are going to mature in the thymus. Okay, so the thymus, red bone marrow, those are considered primary lymphoid organs. Okay, the T cells are going to travel to the thymus. That's a small gland here that's near the heart. Most of that maturation process actually happens even before we're born. So in fetal development, so our T cells are going to differentiate into a couple different kinds of T cells, either T helper cells or cytotoxic T cells are the most common ones. And again, T cells that recognize self are going to be gotten rid of. Secondary lymphoid organs, that's where lymphocytes are activated. So that's places that B and T cells are going to come in contact with the antigen, the parts of pathogens that are going to cause an immune reaction. Oh, and I must not have saved this properly, got some of this chopped off. Lymph nodes, that's kind of the first thing we think of in terms of secondary lymphoid organs. You have about somewhere between 500 and 600 lymph nodes throughout your body. They're all about the size of a pea, so they're pretty small. We can do without some of them, certainly. Sometimes we have to have some of them removed. But what happens there, we're going to see, look at this a little bit more closely, probably next time, but that's where the lymph gets filtered and the B and T cells come in contact with macrophages and dendritic cells that are presenting antigen to them. The spleen, its job is to filter the blood. So where lymph nodes filter lymph, spleens filter blood. And by the way, you have the most lymph nodes in your neck and armpits and groin. Okay. Other lymphatic organs that is on your sheet that's not on mine, tonsils. Okay, those are just big lymph nodes in the back of your throat. Some of us are better off without them. If you have big ones that kind of have nooks and crannies in them that bacteria get caught in, you might be better off without them. Same thing with the spleen. Sometimes if people are in an accident maybe and that spleen gets perforated, you have to just take it out. And people can survive fine without a spleen. They're going to be a little bit more susceptible to certain kinds of infections. They might have to have more vaccines or take antibiotics prophylactically, but we can survive without that. There's one other kind of secondary lymphoid tissue, and that we refer to as the MALT, the mucosa-associated lymphoid tissue. Hopefully that didn't get cut off of your PowerPoint. An example here are the Peyer's patches. Okay, these are some lymphoid tissue that's associated with your small intestine. Its job is to sample what's going on inside your small intestine to test for foodborne pathogens. Okay, and then kind of get the appropriate cells there to fight that. Okay, so that mucosal associated lymphoid tissue kind of lacks the outer kind of tough covering that a lymph node would have, but it functions in the same way. Okay, next time we will look at a close-up of what's really going on into lymph nodes and lymphatic vessels. We'll continue in chapter 16. So have a good day.